I also have the pleasure to have again one of our plenary speakers here, the Needy Bull, who will introduce Gine's talk. So I really wanted to thank you for doing this. Thank you very much. The Needy Bull.
Are you okay, Ines? Or close to saturation? Well, dear colleagues, dear Ines, it is a great pleasure for me to introduce your talk, Ines. Of course, I feel a bit sorry for Pat not being able to be here tonight, but I also feel privileged to have been asked to replace her. Now, Ines is an old friend. Well, I should say, a friend I know from a long time. It's a good start. But I cannot really remember how and when this happened. Actually, I do remember the first time I met Ines. He probably does not. He cannot remember. I was a graduate student. I attended a meeting in the early 80s. I think it was a BSDB meeting somewhere in the UK. And of course, I ended up in the bar late in the evening, having some beers, and at the table next to mine, there were a group of persons talking, two persons in particular, talking a language I had never heard of. Very complex words, sort of a Spanish accent. And there were Ines and Pat, in fact. The language was Drosophila genetics. I was very impressed, and in fact, eventually I was introduced to them, probably by my former boss at that time, was Carl Ilmanzee, who probably had joined the conversation, even though he certainly did not have the equivalent intellectual standard. I couldn't know at that time that I would be, some years later, the neighbor of Pat Simpson in Strasbourg, and certainly not that I would be standing here 30 years later to introduce Gine's talk. But as you know, as coming from the Calvinistic Geneva, I tell myself that somehow it must have been written somewhere. Now, we previously heard a lot about Gine's contribution and how enormous his impact was in different fields of research, and I'm not going to insist on this, including his work with Petru Paul, with Peter Lawrence, with Antonio, which I'm very happy to see here tonight. But let me just say that the paper he published with Ernesto in 1985 had a decisive impact in the start of vertebrate molecular developmental genetics. It was a time when everything came together, along with the cloning of the hermetic complex in flies, in vertebrates, mostly by the lab of Walter Gehring, which I'm sure would have enjoyed being here tonight, the lab of David Hargness and Tom Kaufman. Now, this paper was indeed a key contribution to this revolution. I remember having spent hours to try and understand this paper, my knowledge of this language having moderately improved during that time. But it helped me a lot to clarify the paper published by Ed Lewis in 1978, which I must admit I never fully understood, even though I give it to every single student joining the lab for a journal club in the hope to get some clarification. Now, some years ago, to terminate, I read with interest an interview that Gine gave to a well-known scientific journal. And at the end of this interview, the journalist asked him sort of a classical question one asks to people having reached a certain level of respectability, which is what kind of advice would you give to young people starting their career? And I know there are many people, young people in the audience, who are actually starting their scientific career, and I thought I would give you the two of the answers that Gine gave to this journalist. The first one was, he said, given to him by his mentor, Antonio, when he was with him, is you must sleep with the problem. You need not sleep alone, but you must sleep with the problem. With this obsession, with this sort of profound desire to understand. This is what makes the difference, I think, between scientist and scientist. I don't think it needs a lot of hard work, as was mentioned earlier by Peter Lawrence. It's more probably a state of mind. But the second point raised by Gine in this interview addresses the question that many young people ask. You know, everything has been discovered. There's not anymore any interesting question to solve in biology, etc., etc. And Gine mentioned that, in fact, there is no scientific question which in itself is interesting or is boring. It's just the way scientists approach this question, explain its interest. There isn't anything like interesting or boring science, in fact. And if today we are all sitting here, fascinated by watching the beauty of embryos of all kinds or the precision of developmental processes, it is because great scientists have succeeded to ask the right questions at the right time. Now, Gine is certainly one of them, and this is why I was particularly glad to come tonight here to introduce his talk. So, Gine, please.
Something really bit wrong with the imaging. Okay, it doesn't matter. Well, I don't know what to say actually. First, let me express my deep appreciation of what people like Angela particularly have done to organize this event, and I know the. I know that, that Ernesto and Jordi and Akaimo have been helping her. Also, I really want to thank all the previous speakers for the things they have said about me. Actually, listening to all those laudatory sentences being so nice to me, I cannot help but thinking of Sidney Brenner, who in a similar occasion said that this kind of event is enough preparation from the transition from the present stage of distinguished professor to the next stage of extinguished professor. So I do hope, though, that I'm a long way from extinction yet. Anyways, I'm very pleased to be here. And I want to express my appreciation, my thanks also. First, my family, who is here somewhere. My wife, who is a lighthouse, my professional and personal life is here, Charo, and so is my daughter, Ainhoa, somewhere, and my sons, Manuel and Raúl, also somewhere there. I'm missing my beautiful granddaughters, but they can be excused, they're a bit too young for this kind of thing, but I wish they were there, they were here. And also, I would like to thank the presence of a number of friends from outside the field of biology who have come tonight to listen to me and to be with me tonight, and people from, I cannot cite all of them, there are too many, but people from the physics area, from the chemistry, even from the linguistic area, even from the mountaineering circles. In fact, I just saw our climbing leader, Fernando Martin, who is somewhere there, so I really appreciate that. So I'm really happy and still a bit confused with this situation tonight. And also, before entering the scientific description of things, I would like to thank, especially to a group of people who have been connected with me through my scientific career. Because I want to transform this homage to me into my personal homage to them. Because it is quite clear to me, it should be quite clear to you as well, that if it had not been because of their influence and their contribution, their help and support, I could not have made those discoveries. So much of the credit, whatever I have done, should be given to them. So what I'll do my presentation here is to go through, briefly, through several phases of my scientific career. I will point out some of the what I consider to be more relevant observations, and try to identify the people who actually been connected with it, and to acknowledge their contribution, to give them proper credit. Okay. So, having said that, I show this picture here. Unfortunately, it's somehow it's part of the pictures outside the screen. I don't know why. Anyways, this is a Mount Everest, as a picture I took myself from Kalapata, about nearly 6,000 meters altitude. And for mountaineer, this is a wonderful place. I mean, South Caldera, the summit of Everest, North Ridge, you know, the dream of any mountaineer. But I'm not going to talk about mountains today. But then I want to first to acknowledge a person who is here, because I should say that all those people who have been so influential in my career, many of them actually are here in this room. And the first is Antonio. Antonio was the person who had really brought developmental biology into a new thinking. He did that in the late 60s, early 70s of the past century. And Antonio, this is not working. Antonio had actually been trained in the European School of Development, so to speak. He spent a long time with Harlem, Schubiger, Nottinger, Gehring, all those guys, Bryant. He had this training in classical developmental biology, Drosophila, a typical European thing, transplantation analysis, the development of concepts like determination, transdetermination, that kind of thing. But then, after having spent maybe 2 years or so with Harlem in Zurich, he went to Caltech. There, he was exposed to the influence of the great school of Drosophila genetics in the USA. I mean, people like Sturtman, friend of his, Ed Lewis, and Linsley, and others. And Antonio was able to assimilate the two types of thinking, and we came back to Madrid, and he made a synthesis, so putting together this school of Drosophila genetics, the genetic technology, genetic thinking, typical of the Americans, and the developmental thinking, typical of the Europeans. And then this is a conceptual basis of what I could call the Spanish school, which was founded by him. We could call it the genetic of development, the genetic of development of Drosophila development. So I had to acknowledge he was my thesis supervisor. He has a key role in my career. Lately, he has got some funny friends. The guy in white is a Catholic saint. You probably know that. So I guess Antonio makes friends with him, because he's expecting when he will be helping him to get into heaven when the time comes. So I hope it will be a long way from now. Anyway, so what Antonio had done, among other things, is to develop really powerful technique of cell lineage in Drosophila. You know, to an audience like this, I don't have to explain how important cell lineage studies are. But what Antonio did, he published some key papers with John Merriam, is to use the mitotic recombination event to study cell lineage. The idea is that you irradiate non-lethal dose, you can produce a certain amount of mitotic recombination between cells in the G2 state. I don't want to go into the details, but the thing is that you irradiate these cells. The time of irradiation will mark the beginning of the development of a clone. It will be, you can tell the cell of the clone after the mitosis, because they acquire a genetic marker. So in irradiating imaginary discs at different times of development, he could have he could study this life story of the different discs at different times of development. Could it be possible to move the image a bit to the left? I don't know if somebody's in charge could do it, because I guess I'm going to miss some of the things. I don't know whether that could be possible. ¿Podría ser posible que la imagen se volviera un poquito más hacia la izquierda? Es que estoy perdiendo los detalles de... Is any way you can... In the computer? Well, okay, you come here.
Lo conseguimos, no es fácil.
Sí, pero es que estoy perdiendo desde aquí para allá, que es una pena.
Espera un momento, a ver si lo consigo abrir. ¿Cómo hago un double clic en todo?
Ah, es que no funciona, tienes que funciona con el ratón. Con el ratón sí funciona. Ay, no, parece que ahora está bien, ¿eh? Sale de ahí. Puedes sacar con el ratón, ¿eh? Es que la pantalla está rota. Oh, see what happens now. Oh, wow. Oh.
Tengo que cambiar de pantalla. No pasa
¿Cómo?
nada.
It's confident.
Hizo el ratón.
¿Cómo? Ah, tienes ratón.
A ver ahora. ¿Dónde está PowerPoint? A ver si funciona ahora.
Dale a ver si... A ver si...
Sí, pero es que tengo que darle... Joder, mover esto aquí.
Ahí, dale, ahí, eso ahí.
Ah, está.
Ok.
Espera. No vamos a poder hacerlo así.
¿No?
No. Vamos a hacerlo de otra manera. ¿Cuál queréis? El 512, ¿no?
No sé. Yo como nunca le toco porque siempre suele estar bien. Bueno, si no podemos tampoco pasa nada.
No pasa nada. Es que no sé qué tamaño tienes que coger. ¿Qué tamaño tienes que darle?
Es que...
de resolution.
Sí, eso es lo que estamos haciendo.
Pero tienes que en la alineación tienes que poner reunir pantallas.
Sí, están unidas.
Vale, entonces ahora en la resolución de la pantalla hay dos pantallas, la LCD de color y la pantalla VGA. Entonces, esto ponlo en 1024 x 768. No, esa no. Sí, es la resolución de la salida, sí.
Ahora.
A ver, abre la presentación, a ver qué pasa. Aquí.
Oh, very good. Oh. Okay, well, thank you very much for your help. You have saved my presentation. Anyways, this is a typical experiment that Antonio and Maria were doing, and they were collecting data from different clones in these different moments of development. Okay, so I was a graduate student at that time, and I was with my colleague Pedro Ripoll, who was another graduate student. Pedro is very good in collecting mushrooms and things like that. Also, he's a really brilliant geneticist. So he and I together developed a new method of essentially to alter the growth rate of marked clones during development. This, I say this is a faithful paper for that reason will be apparent immediately. The first thing we did was what is called the minute technique. So generating clones of cells with higher proliferation rate than surrounding cells, okay? This thing has already been mentioned several times. Essentially, we managed to produce a marked cell which has a proliferation rate higher than their neighbors. So we found this cell could make huge clones. You compare these clones produced by this technique, cloning pink, compared with the conventional clone. So those clones were gigantic, okay? And that was essentially the technique that led to the discovery of compartments. Essentially, these big clones actually delineate the compartment for you. After having looked half a dozen of these clones, you see the anterior clones will stop here, in the middle of an invisible line. Or else it could be posterior, but stop in the middle here, in the same place. So you realize immediately there is something special about that line. That turned out to be, that's an anterior-posterior compartment boundary. So, and then we published, together with Antonio, this Nature paper, and we described compartments for the first time. Later, people or work by other people, including especially Peter Lawrence and myself, show that this compartmentalization is not only specific to the wind disk, but it affects the whole of the body. So the subdivision into anterior and posterior compartments is a major feature of the organization of the body of the fly. Okay, okay, that's what I had to say about compartments. Compartments has become a key issue in Drosophila development from that moment. Now, but in that same paper, Pedro and I were kind of vigorous and young at that time, so we had to do something clever, not so straightforward as the other thing, but to use a genetic trick to generate clones with low proliferation rate. I had to say that I actually think to believe that Antonio himself suggested that experiment, but I cannot remember. It's such a long time ago. So if he did, I'm very grateful to you, Antonio, for the suggestion. But might have been Pedro. I don't know. I believe it was Antonio. Anyways, the thing is that we use translocation called SQTS2, and what happens with this translocation is that you have a piece of the second chromosome carrying a doses of minute. Minute is a gene encoded for ribosomal proteins, okay? So, and the reason why the minute fly grows slowly is because they have limited amount of ribosomal proteins, okay? In this translocation, they have a normal, the translocation is normal dose of ribosomal proteins, but in the progeny of the translocation, you will see that a fraction of the fragment of chromosome 2 carrying that minute gene is now in the first chromosome. So it interchanges genetic material. Then you do a recombination, mitotic recombination there, you will find, in half of the occasion there is a recombination here, you will get a cell which only contains one dose of this dose of the minute cell. So you see, you generate a cell which contains only one dose of minute instead of having the normal 2 doses of minute. So that because that cell has relatively few chromosomal proteins, it will grow slowly. Now, we expect this cell simply to grow slowly, but what we found is that as compared with controls, these slow dividing cells disappear. They are eliminated. We couldn't see them. And then the important thing is these slow dividing cells are perfectly viable. We could make flies of that genotype, which are perfectly normal and fertile. Yet, you have those cells with that genotype surrounded by normal cells, they are eliminated. This is what we call this phenomenon cell competition, because we assume that we thought that somehow these cells could not compete with other more active, energetic wild-type cells. And I had to acknowledge here, this is the phenomenon cell competition, which is an interaction between two types of viable cells. But that led to that's the key thing about cell competition. It is an interaction between two types of viable cells, but one of them is, because of the interaction, one of them is going to die. Okay, I had to acknowledge the contribution to this phenomenon. Pat Simpson, who was supposed to be here, but she is unwell, could not come at the end. But she has made important contribution in the early phase of this compartment idea. In fact, the paper that she and I published in '81, it specifically said that it must be emphasized that the minute cells are only unviable when are growing next to non-minute cells. The process of cell competition therefore relies upon cell interactions. And that's the key issue about cell competition. Okay, this is Pat. Now is a member of fellow Royal Society. She was then, that's the time we published this thing. Okay. Now, that was the work I did in Madrid, partly also in England. But then I moved to the next phase of my career when I met Peter Lawrence. Actually, I had met Peter Lawrence before. He already described those events. But I was doing some inconsequential postdoctoral work in Oxford. Then I was not so happy. But then that time, Peter Lawrence asked me to come to the LMB in Cambridge to give a talk. And that was really an important talk in my life because the talk was supposed to last for about 45 minutes or 40 or 50 minutes. And the talk in my broken English actually lasted for 1 hour and a half, or even nearly 2 hours. And I was continually interrupted by this guy here, Sidney Brennan, with huge eyebrows, asking a lot of penetrating questions. I was so, in a way, I was so happy, but also I was so afraid of the guy. Anyways, I guess that I caused a good impression on Sidney and on Peter. And 2 weeks or 3 weeks later, Peter sent me a note saying that if I wanted to work at the LMB, I could come. So what I did. And then I started the collaboration with Peter Lawrence, some few pictures. Peter was very young, many years ago, and now it's more older picture. We have been working together for, collaborating for over 40 years or something now, and published 22 papers, as he said. I had to say, the papers I published with Peter Lawrence are probably among the best papers I ever produced. I'm really, proud of those papers. I think as he mentioned, I think it's quite clear, quite true, that we kind of complement each other. I don't know how, but we sort of complement each other. I mean, he made the point that we are lazy. That probably he's talking about himself, not about me. But I think he's confusing hard thinking with hard working. And geneticists are usually hard thinkers, maybe not so hard workers, okay? Anyways, I cannot go into describing all of these papers, but let me mention something which already mentioned, and that is the biological significance of compartment. In fact, they were received with quite skepticism by the people who knew. For example, we attended a meeting at the Royal Society in 1976, and Sir Vincent Wigglesworth, is a really great guy, this is a very, very important guy, who said, I'm sure Lawrence and Morata are right, and this is very fascinating, all this is very fascinating, but why this compartment for? I mean, you see, the compartment is a particular, the best compartment, the best known one, is an invisible line in the middle of the wing. I mean, not associated with any morphological feature of any kind. I mean, what's the meaning of that? I mean, it was, so many people share this view, okay? Of course, we have other reasons for it, because our Bythora work to think there's something genetic about it, but people didn't know. And then what Peter and I did, essentially, was to provide a genetic basis for compartmentalization. And that we did that with engraved work. I had to say, that previous to our work in this Nature paper in 1975, there was a paper by Antonio and Pedro Santamaria, was 72, Antonio, I believe, doesn't remember, I do. And they have shown something very interesting. In this engraved wings, it's a leaky mutation, the posterior compartment, the posterior region of the wing is very much like the anterior one. The bristles are similar, the patterns kind of similar. So it was a kind of good case for something involved with the AP compartmentalization. And then what Peter and I did was first to do a clonal analysis of these engraved wings, and we showed there's no lineage that you could draw. While in the wild type, no lineage boundary, I mean. While in the wild type wings you can make it, a very straight line. In the engraved wings, there's no way you can make such a line. So there's no clear lineage border between them. And the second experiment, a really important one, is that when you make clones of engraved mutant cells in the anterior compartment, nothing, absolutely nothing happens. It's perfectly normal. However, if you make the clones in the posterior compartment, the clone is transgress the AP boundary, it make anterior pattern. So that showed that the engraved is a genetic marker of the posterior compartment. It is expressed in the posterior, but not in the anterior compartment. Okay, and that actually was a big thing about it. I mean, really important information to understand the genetic basis of compartment. And he and I published a big review together, linking homeotic genes and compartment cell determination, explaining, essentially we provide a genetic doctrine of compartments in those papers. Okay, now I had done my work in Bythora, my PhD in Bythoras, and I was aware of something that people were not, and that is that these lineage borders we could see using the minute technique actually corresponded very well with the mutation, with the phenotype of certain mutation of the Bythora complex. So in this bithora mutation, the anterior heart is transformed into an anterior wing, but the transformation is defined by the AP border, and the same goes for the posterior compartment. So that suggested these lineage blocks has something to do with gene function of the host complex. Okay, and then so I was always aware of that. And then when I was in Cambridge, we had a visit of Ed Lewis. Ed Lewis was a great guru of bithorax. Okay. But the only one, only him was working on bithorax, actually. But we, of course, were very interested in the issue of the bithorax complex. So Tom Comberg he's here, but he also there. Also, this is myself, Peter Lawrence, Mary Bounds, and Tom is not in the picture because he actually took the picture. And then he's Ed Lewis here. He gave a totally incomprehensible seminar. Nobody understood a single word. However, we have been practicing in his, we call Lewis diabolical nomenclature. He had developed a nomenclature which nobody can understand. I mean, we spent about 2 weeks practicing about these ABCDs and T1, T2, T3, T4. I mean, you cannot explain this to you. I mean, it's just, but it's so difficult that as I say, we spent 2 weeks on it, and I used to call in the mornings and say, okay, Tom, small a over big C in T's. And then he would think for a while and say, okay, that would be slide T1, slide T2, partially transformed T4. Okay, right. So we really got very good at this. So Ed came, and during this discussion, I remember it very well, I said, Ed, I do not really understand this transformation of D over E. And he said to me, what are you talking about? And I said, well, you know, nomenclature, this 2 over D, this T1, T2, T3, no. I abandoned it. It's totally useless. Nobody can understand it at all. So that we have spent 2 weeks wasted, actually, 2 weeks for nothing. But we were very good at this. So sometimes we play to it. Anyways, but then because Peter's influence, he managed to convince the editors of Nature to publish this paper by Lewis, putting, as I say, publishing all the information there was about bithorax. The paper is totally incomprehensible and mostly wrong. But there is something important in that paper, was the phenotype of the P9. P9 is a deletion of all the bithorax genes. And so this is wild-type larva, different segment, you can tell, different kind of segment. However, if a larva which lacks all the bithorax genes, all the segments develop alike. All the thoracic and abdominal segments are the same. So what happens when bithorax gene is missing, is the entire diversity, morphological diversity of the larva and the fly, what we saw later, from the thorax to the abdomen is completely lost. So it's not the segmentation, as he said in the title, that's also wrong. It is the way the segment developed with this ROM in the bithorax complex. But then he proposed that there is a minimum of 8 genes in charge of this deformation of individual segments. You think this is quite natural to think, you think flies is divided in segments, so you tend to think, okay, your segment is something you see so well with your eyes, so maybe genes should be working, should also recognize segments and important anatomical portions, okay? So, based on the phenotype of certain viable mutations, like the UBX already shown, you see, in this mutation, only one segment is transformed, the others are normal. And this mutation is BXT. You see 4 legs. That means the transformation of one abdominal segment into a thoracic segment, making 4 legs. So all these mutations seem to affect only one segment. So also, the logical idea was presented by Ed Lewis and by Gary Struhl, is that there should be as many bithorax genes as segments or metameres. I think Gary's model was much more sophisticated than this is Gary's model, this is Lewis's model, because he had to interpret some results from our lab and also from his own lab, showing that the limit of the bithorax mutation is not a segment boundary, but anterior-posterior compartment boundary. But anyways, so the auto-register model, he's the normal model. Gary's model was an advancement over Lewis's model, but not entirely correct in my, because essentially I thought that something was not totally wrong with this model, from some observation I had made previous to this. So when I came to Madrid, having finished my work in Cambridge, I was so incredibly lucky and fortunate to enlist two incredibly gifted students, Ernesto Sánchez Herrero and Jordi Casalua, plus Rosa González, who was an incredibly good technician. So that's the group we started. I really was fortunate to have these three people working with me, because much of the thinking and certainly most of the work is from them. And then we decided to look, to do a fresh approach to the bithorax complex. How many genes are there and how they function? So we published a number of papers, which I'm not going to describe here. And also Anna Busturia also joined us at some later stage. And then essentially we came to something very different from what Ed and Gary has suggested, and that is the bithorax complex contains only 3 genes. That pure genetic analysis, classical complementation analysis, just 3 genes, not 8, not 9, 3. And then when we put together the Lewis Strel model, you see the all genes per segment or per parasegment, and the Madrid model is only 3 genes. Of course, you have to explain. I mean, it's not too easy to swallow, because I mean, how a single gene can make all this, the morphology, all this, so that's just a complicated thing. But then we had a lot of trouble from the American people, because nobody actually did not believe us for a while. But we were lucky, in a way, well, to say that the same 3 gene domains could be applied for the adult fly, as indicated by this color code. But then, shortly after we published this paper in Nature, there was a paper, I think, in Science, showing that there are 3 homeoboxes in the bithorax complex and 3 transcription units. So that settled the issue, and it turned out that we were right. So this is a meeting in the Cold Spring Harbor. A lot of important people here. Well, this is myself, but not important. You see, this is the Nidobull here. Dave Hovnes, Kafatos, Jerry Rubin, Tom Conver, see? Stuart Kaufman, Sigmund Benzer, Jim Watson, Walt Bender, Bishaus, Janus Lane, John Gordon, Peter Lawrence, Ed Lewis, Jacob, Brenner, Louis, I don't remember who is this guy. An important guy. Anyway, so here, I think the question was settled. There are 3 gene bithorax, and all of these, the segments are made by specific activity of cis-regulatory elements, okay? Now, after that work, we followed for a while, studying this important issue that was mentioned by Denis yesterday, about this idea of collinearity and functional hierarchy, something that a functional hierarchy in a way was discovered by Ed, but was actually really well presented, well elaborated in a wonderful paper by Gary Strull, where he showed the individual function of the bithorax genes, and showing that there is a functional hierarchy. The most posterior acting genes are functionally dominant over the anterior one. But look, see, the important thing is that you see the things are collinear. You see the expression in the fly on the humans, and you see the more 5' genes are expressed more posteriorly, and the more 3' genes are expressed more anteriorly. This is a collinearity that nobody really understands, except that Denis is the only one who understands, okay? It's a major, major issue. And then we discovered, well, this guy also was very lucky having a Kaimo here. You see this white back here? I think even sleeping, he wears this. Whenever since I have known him for 20 or 30 years, always wearing this kind of funny way. Anyway, not today for some reason, but anyways. He was really forced in the lab. And then we published this paper with Gary, which looks very much like Richard Feynman here, actually, and Walter Gehring, which unfortunately died of a car accident some few months ago. Gary, I'm sure, remembers the difficulties we have writing this paper. Anyway, with the phenotypic suppression, we provided the basis to understand this functional hierarchy, why the more posterior genes actually are dominant over the more anterior ones. It's not a transcriptional control, as it was thought to be. It's a question of protein protein-protein interaction. Anyway, it was an important paper. But you see, Denis has published a paper in development showing the same thing for the mouse genes. And actually, we got together, we published together with Denis a paper showing that the mouse Hog genes, if put into the fly, actually show this same phenomenon, phenotypic suppression, indicating this a general universal feature of Hog genes. And also, Anna Matthias in the lab showed that in fact, phenotypic suppression can be used in normal development to turn off certain genes, which even though they are transcriptionally active, the fly does not want them to function. So the function is stopped by this phenomenon of phenotypic suppression. Okay. Now, having been for quite a long time in that, I had to look for more genes which are of, important in development, more Hog genes or developmental genes. And then with the help of Manolo Calleja, who is also here, we developed this yellow method, which is actually very clever, what he and Manolo did. Essentially, it's a geneticist dream. You don't have to do much. You cross males and females and look what happens after. So you don't have to manipulate anything, just to look flies. And then, essentially, you can generate a large batch, a number of new Galfo lines, and then and test them with the US yellow transgene, and say everything is yellow in the fly except the gene in which the P element has inserted, and then the gene is active in the wing or in the antenna. You have this, they have a black antenna and black border or whatever, okay? And we essentially found a number of new functions for known genes and a new gene that work in the adult cuticle. And for example, Willis appears in the adult cuticle like that, you see? And Odd Skipper appears like that. And Panier appears like that. All these led to papers which are not going. And by something else, I want to show this an allele-specific gene, because I have not mentioned before, and that is when, I think maybe go back just a second. You see, the Bythora genes is playing this part, this part, this part. This is a black dot here. That's the analia, okay? So that part of the adult fly was not accounted by Hog genes. And now we found this gene, we cloned the gene, it turned out to be caudal. Gary has been done a lot of work on that, but he is not aware that they have an adult function. And this is how caudal looks in the analia. We label all the analia. And Eduardo Moreno, who is somewhere, he and I published this paper in Asia showing this is, so to speak, the last Hog genes, the caudal gene. And now we're quite happy that we can account for all the body parts, including the caudal. Okay? So one of the many contributions of the yellow system that Manolo developed. Now, in the early 80s, the competition re-emerged again. And that was thanks to Eduardo, who really, he has done the work in the lab, the analia work, but then he really got involved in this competition business. So he wanted to work on it. So we published, in collaboration with Connie Bastler, a paper, essentially, the genetic system is just as we had done many years before, Pedro and I, essentially the same translocation, but now we can look at the adult, at the imaginary disc, we can label clones in vivo in the disc. Essentially, we found that these clones with slow dividing cells, they are perfectly viable. The slow dividing cells are the double green ones, and they, after 24 hours of life, I mean, generate the clone, they are alive, but at 48 hours, they have disappeared. The double green ones have disappeared, only the control, which are black. Okay? So we thought, okay, maybe 36 hours should be dying. In fact, they do. That's what happens. And then we, with the apoptotic marker packer, which is a marker of the JNK pathway, which is an apoptotic pathway, we can see all these slow dividing cells are labeled with packer. So they're actually dying. So the important thing here, apart from the effect of DBP, I'm not going to come into, is that this the cell competition functions by apoptosis, and apoptosis is mediated by the activity of the JNK pathway. Okay? That I know I explained very well how we got into this. When we present the things, we try to present the things in a kind of logical way. But she clearly explained, often we do things which, not because any logical thing, but because things happen, you find them unexpected, you do things you do not want to do originally. Anyways, she presented it beautifully this afternoon, is that you have produced a lot of apoptotic cells in the undead cell paradigm. So you induce a lot of apoptotic cells, but do not let them to die using the P35 protein, and essentially those undead cells do not die, but they produce tumors, hyperplastic tumors, okay? And she showed for this case in which these green clones are clones of apoptotic undead cells, a lot of increase of proliferation in the region nearby, close to it. So these non-dying cells are sending some kind of signal to the surrounding cells to proliferate. Okay, this is another way to say. So the important thing here is that these apoptotic cells are releasing mitogenic signals. And that's actually very important. Some later papers, we actually show that that's dependent on JNK directly, that is directly responsible for activating Willis and DPP and producing non-autonomous proliferation. Okay? That leads me to the last part of my talk, and the people are having here in the lab now is Angelica Cantero. I should acknowledge her here. She's around, but she is the lab manager. She's the one who does all the things in the lab. She knows everything, so we cannot do without her. There's a whole list of people. I cannot go into each of them. And this Natalia Spiazzo is missing in the main feature, so I put her here. But she's also, of course, an important member of the lab. Manolo is here. Anyways, what this work on apoptosis led us is to study the connection between apoptosis and tumors, and cell competition and tumors. And then we have been doing some work using some of these oncogenic mutations in Drosophila. There are many of them. Mutation, all these pathways can lead to tumors. But I'm going to tell you briefly about RAF5 and LGL. RAF5 is involved in endocytosis. It's a defect in endocytosis, so it produces a lot of problems in the cells. And scribal lethal giant larvae, they affect the apicovasal polarity of the cells. Very important to keep the epidermal cells to have the appropriate polarity. And when they are mutant, for example, I should say that all these genes are known in humans and produce tumors in humans. So we like to think that we find something of interest. One day might be applicable to humans. So if you look at, for example, LGL mutant larvae, these wild-type larvae, they're gigantic larvae. They will not make an adult. This is why they're called lethal. But you see, they have these hyperplastic tumors in the brain and also in the imaginal discs. But normally, tumors do not appear because a person or a fly is homozygous mutant for something. Tumors appear because a small group of cells become oncogenic and are able to colonize or proliferate and colonize the rest of the tissue. So what we decided to do is to make clones of oncogenic cells in a wild-type tissue and see how these tumors develop. Okay, the clones are in green here, and you can study what happened to these clones, how they develop, how these oncogenic clones in the wind disk. And we found that, to our surprise, about some few years ago, these oncogenic clones are eliminated. They disappear. And they disappear from the tissue by cell competition. That's all the features of cell competition. They're eliminated by JNK-mediated apoptosis in the periphery of the disc, of the clone. It's perfect, just cell competition. And this is for LGL, but RAF5 mutant. It's the work by Luna Ballesteros, who is somewhere here in the meeting, and showing that these clones are also eliminated. And you can see the elimination. In the elimination here, you can see the caspase activity, which is indicated the cells which are dying, are in the periphery of the clone. But inside, they're still alive, but then eventually they will be disappearing as well. And then we proposed a model with Javier Menendez, I know, Pérez Garijo, few years ago, is that because how is that the tumor cells are normally eliminated, but how is that sometimes they produce tumors? And for a number of experiments, we came to the conclusion that there is a microenvironment thing. So there is a group protection mechanism. So you make a large patch of mutant cells, and then there are a lot of cell death in the periphery of the tumor. You see, the red here indicates dying cells. But inside, the green means healthy cells we can divide. Okay? And this is you only look at the red channel. Okay, this group of cells, is too small in all the die. So we came to the conclusion that this is a group, it's a group protection mechanism here. So we tested the hypothesis recently, essentially transforming the entire domain, the SPAL domain, from non-tumor to tumor using the GAL80 system. That doesn't really matter so much, but we have the means to have a normal with this here, with this domain, and we can transform this domain from normal into tumor. And the time we do the transformation, the SPAL domain contains 400 or 500 cells. You do that, and then this tumor develops and is not eliminated, okay? And also you can see there's a ring of dying cells protecting the inside of the green cells that are healthy. The dying cells are also green. You can tell it's faint green here. They have been eliminated. Okay? But we find something really unexpected. I'm about to finish now. And that is that we look at the border of these clones, and we found a correlation between high level of BRDU, that is high level of proliferation, and high level of apoptosis. It's indicated here. So that didn't make sense in principle. So why is that? So this is because this is the area of intense cell competition. And then we come to look at this image. You see, this is a border of one of these tumors. In blue are the cells which are dying. And then, okay, good, thank you. And then you look, the red stain is BRDU. That means it identifies cells which are dividing. You go, as you see, the dying cells, and very close to them, they are cells in active division. Okay? So that suggested that this dying cell was signaling proliferation to the neighbors. In fact, that immediately reminded us of this phenomenon that I know and others have described some few years ago. That is the apoptotic cell, and these apoptotics are actually sending proliferative signals, and the growth of the tumor might depend on the presence of those signals. In fact, we immediately decided to look at this and to check that idea, and then this is the idea was that this J&K signaling is responsible for the Winless NDPP, and you can see the tumors, the high level of Packer means high level of J&K activity, and and then you can also see the Winless. I think this is Winless is really active in those tumors. Now, if now. No, we're going to go to the next one. Now we prevent activity of J&K, and then you can see now essentially a Winless expression, it goes back to normal in comparison with this. But not only that, now you prevent apoptosis completely by making this transformation in the drug minus mutation, in which there cannot be cell death. You see the tumor doesn't grow, as indicated here. So in the absence of cell competition and apoptosis, the tumor doesn't grow. I think that's a really major finding, because it has certain implications that I will be telling you in a minute. Because the general conclusion from this work we have been doing lately is that cell competition is a powerful mechanism to identify and eliminate tumor cells. That's the normal function, and the development of a tumor that requires the tumor cell generate a protective environment. Otherwise, they will be eliminated by cell competition. Now, once a tumor is growing, cell competition reverses role and now actually stimulates tumor growth. So let me check. I mean, how much of this is relevant to human to situation in vertebrates? Okay. First, there is a recent paper by Miguel Torres, who is somewhere here, in Nature, showing the the cell competition actually does occur in vertebrates. It plays a relevant role in early development. So cell competition is not a specialty of Drosophila, but it also occurs in vertebrate, mouse development. Okay, and also there was a previous work in gastroenterology indicating the cell competition is involved in liver regeneration in rats. Okay, so it's not a specialty of Drosophila, but also we say cell competition is a powerful tumor suppressor mechanism. There is a very recent paper in Nature, some 2 months ago, that cell competition is a tumor suppressor mechanism in tumors. I cannot go into details, but it's a very beautiful paper. I suggest that you read it. But essentially, what it shows that cell competition is involved in the elimination of late progenitors of the T-bone of the T cells. When the new cell coming from bone marrow eliminated by cell competition the previous one, so that's how the system works. If they don't do it, and they will make a tumor. And finally, we say that in a growing tumor, cell competition apoptosis stimulates the growth of the tumor. In fact, there's a recent paper in Nature Medicine showing that this is the basic information here, is that you take tumor cells and you irradiate a group of these tumor cells with heavy dose of radiation, which make them essentially dying, and then you mix those cells with normal cells, non-irradiated cells that produce a strong repopulation of the tumor. And that's mediated by a growth factor, in this case, PG2, is prostaglandin 2, just like as Winders in DPP. So essentially, the same system we are finding in Drosophila, we can generalize, or at least we can think that is also happening in vertebrate, is to indicate the important role the cell competition might have. And there's a warning here, the cancer patients are often treated with pro-apoptotic agents, radiation, chemotherapy. In some cases, this may be counterproductive. So I think to think about. I mean, having talked to several doctors, I seem to have a problem there. Okay? So that's stop here. Really thank you for your attention, and I would like to thank all these guys, and maybe some of them might be missing, for having contributed so much to my career. I had to finish by saying that without having been associated with them, my career could have been completely different or non-existent at all. Thank you very much for your attention.